Hello everyone. In today's tutorial, we will talk about how to download a genomic coordinates of a specific gene. And the genomic coordinates may include the exonic regions and the intronic regions. So this challenge comes for the bioinformaticians or biologists that if they have multiple gene sequences and they want to know a specific exonic region or intronic region, and it's difficult doing it manually. And although it's easier for one gene, you can go and you can type the gene name on ensemble UCSC or ncbi browser, and you can see that from where it starts and where it ends. For example, if I show you here, if I go to the ensemble genome UCSC genome browser here, if I click the browser, and now what I do here is that, for instance, I go to the browser, and in the browser I type here some specific gene name, for example TP53. So if I click it, then and search it, it will give me an output here that this is the TP3 gene. There are multiple transcripts of it. So there's some upstream region, then the exonic regions are in boxes, and the intronic regions are in arrows. So the big intronic part, but the smaller exonic part. But you can, if you click it, you can go to the detail that the sequence and so on and so forth, everything. But usually it gets challenging when you have multiple genes. So how you can do it? The thing is, there's a very beautiful feature in UCSC, that is table browser. We just click the table browser, and apparently the gene coordinates which I selected are chromosome 17, and this is the TP3 region which I searched. One way is that for example, if I am looking for this position of the TP3, that how many coordinates, how many exons are there and how many introns are there. So first you have, like, obviously this is mammals, and then human. I've searched for the humans, and then in the groups you have to select the genes and gene predictions, and it's already selected. But you should do it if you directly go to the table browser. Then CBRF seek is the most commonly used track. So or I can use the gene code for the gene information. The table I want to get is the known gene table, right? And now what I have to do, that this is the chromosome coordinate. One way is that I will tell you the other one, that if you have to upload multiple coordinates. One way is this, that these are the positions, and I want to have the bed format, and I want to download the TP53 sequence dot text and get output. So here it will now ask me that, do you want to have the information of the whole gene? Do you want to have the information of the exonic regions or intronic regions? Whatever is the case. So first I download from the, for the whole gene. You can see that this TP3 gene I've downloaded, it has the all the coordinates, right? So whatever information it has, the start and yeah, and what is the length, and how many exons, introns, and everything is here. So it's just give you everything. From here you cannot understand what is the header. So for that, like for instance, you can go here and you can see the details. I'll show you in the back, but in the back, when I'll do go back, I will show you the description. But so it has everything here, right? But if you want to download the exonic regions only for that gene, you can say that go and get ped for only the exonic regions. So it is now giving you the exonic regions. For example, if you see here, yes, so now these are all the exons of multiple transcripts. So obviously, one gene has multiple transcripts. So chromosome number 17, start and end position of the transcript, right? And sorry, start and end position of the exon, and this is the transcriptome ID. This is exon number 6. This is exon number 5. So there are total number of exons In TP3 are 0, 1, 2, 3, 4, 5, 6, 7. So 7 exons. And for this and this multiple exons for this, you know that one gene can have multiple transcripts. So TP3 is a very big and very famous gene. That's why I search here. Now, if you want to get the intronic regions, you can just click the intron. Then you can download the intronic part as well, right? So these are all the intronic regions of those the same transcripts I've shown you before. Now this is one way of doing it by 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 searching for a specific gene, right? For example, here you can see that this is the description of that file I showed you. This one, the first one here, yeah. So one first is the chromosome name, and then the transcript and start, end, and the coding and start, and then all the exonic start regions and end regions, protein ID, and so on and so forth. So you can match it with the output of the file. One more thing is that it's for the specific region. But for instance, I want to upload a list. So here you can see that there are some examples like I want to submit multiple gene IDs, and for example, these are three for transcriptome IDs, and I want to search them. Then you can go here and you can put the list, and then you get the I just intronic part that you give you again. But for now it's because we are getting the we are only looking for this coordinate. But if I want to define regions, so that's the list is already uploaded. Yeah, here you can put, for example, this is one information you get from here, that you can have multiple coordinates, and you can upload a list of these coordinates, and it will give you the output of all the different genes you want to look for. Okay, so I hope that would be helpful, because for it's always very challenging to know that what are the genomic coordinates of a specific exon, or what are genomic coordinates of specific intron of a gene or multiple genes. So I hope this tutorial was helpful for you to find those coordinates and the annotation of those genes. Thank you. Bye bye.